Hello, keeping. The next, which is dihybrid cross, I have told here Mendel has taken the round seeds and the green seeds. Round is the dominant here. Round is the dominant here, which is capital R, capital R. Green is the small, which are smaller, smaller. So, which are yellow, which is a dominant character, I have drawn here the capital, and wrinkled I have drawn the small y's. So, the gametes, when they are separating in there, they have come capital R and capital Y and Y. The cross which has done by these, the F1 generation has come, which is heterozygous chromosome, one capital and one small. That is why these are, because dominant character is capital R and this one, the seeds which are came here is the P seeds which has come is round and yellow. So further F2 generation, what is that we have to do now? The dihybrid cross, which is done by two characters, which are these. So these, when like tabular form only, you can do calculative part, and here what are the different things we are going to do one by one here. So, and the result also we draw. So here the first one, when you are dividing this, the first one which has come capital R, capital R, capital Y. Okay. So same thing with this Y and Y, and again capital R, small r. See how I am, this one and this one, I am doing multiplication like that. So carefully you see this. Even here one capital R, small r, capital Y, small y. What we got here, that is K. This is the, I am writing the phenotypes which are present. When they are doing same thing, this one with those, this one is coming. Then capital R and capital R and small y's, those are coming. Even here capital R and small r and this. And the last one which has capital R and small y's both. Even I will write the which seeds we are going to. Even when they are crossing, this one capital R, small r and capital Y, small. And when this and this is what? Both are capital Y's. Even the third one, which are these two. And the third one which is capital R's and capital Y and small y. And the last one which we are doing, almost all, both are recessive character only are coming. No, here it is, I have to do R, this one, right? So obviously the capital Y, what we have got there, like that only. Again, when they are doing this one with this and this one R and a small r and a small y's both. Okay. The next one is, we are going to do like this. The last one is the complete recessive. So now we are going to see that the ratio here it has come, you can write it as round and yellow. See the here complete round and yellow character has come. Here again it is round and yellow. The seeds which are here, this one round and yellow. The numbering we will write. This one is also R dominant. Again this is also round. When Y is, that time this is yellow. This one again both capital, so it is complete round and yellow. Here round, the first character which has come, but small y's, because of that, that is wrinkled. Okay. So here also round and again yellow. Here round and wrinkled. Okay. carefully see, write like that. When you are forming in exam, if it is coming, you have to do the diagram like this. So the diagram, again the round and yellow only, many. So total 9 might come, round and wrinkled here. Okay. Here it will be green. First one green because the smaller which has showing this. The capital wise when it is showing, suppose here homozygous, this is homozygous. So it is yellow only here. So this one green only and this is yellow. Here again round and yellow. Okay. Here it is round and small wise. Small y is showing wrinkled. So it will come wrinkled. Okay. Here again it will be green and yellow. The last one, only one will come green and wrinkled. This is the poor recessive character which has shown. So the phenotypic character you can draw like this. The phenotypic ratio and genotypic ratio are so you can write the character which are showing, you can count the round and yellow. How many? 1, 2, 3, 4, 5, 6 and 7 and 8. No. 1, 2, 3, 4, 5, 6, 7 and will come totally the 9. Round and yellow will come 9 is to the 3 is to 3 is to 1. The phenotypic ratio. The phenotypic ratio is this one. So which is here the mixed 3 and this is completely green and wrinkled. The last one which has there, that is green and wrinkled here. So center one which is 3 are round and wrinkled and round and wrinkled and green and yellow, which are 1 and 3 and round and these 3 are this one. So here round 1, 2 and 3. All others are here capital R. So you have to do this one carefully. And another ratio, the genotypic ratio is 1:2:2. The genotypic ratio which is showing here is 1:2:2:1:4:2:1:2:1, which are showing these all complete R's, and here complete R and Y, this mixed which is showing heterozygous condition, again 2, and these characters by these counting you can write this. This has phenotypic ratio. So no, this is genotypic ratio. In the exam they ask which is the genotypic ratio and which is the phenotypic ratio. You have to count this and write how many are these, like that. Even capital R's and Y's are here, it is showing. So next I will move, hope you understood this. Next I will move on to in this Mendel's law, dihybrid cross and monohybrid cross, everything I have explained. Now I will tell the several which is left over: incomplete dominance, codominance and complete dominance. There are another definition which is there. Those are now it is next one is incomplete dominance, which I will show here. The incomplete dominance. Write about incomplete dominance, they say. So, and this is also called as semi-dominance or half dominance. Half dominance. Okay. Even the definition of this, suppose if you observe this incomplete dominance, he is under 4 o'clock plant and also called as Mirabilis jalapa, which is a Mirabilis jalapa is a biological name. Then another name is snapdragon. In snapdragon, Mendel has did several experiments, so the result is different. See which 4 o'clock it blooms in the evening, pink color. When he has taken, now it is pink, red flowers he has crossed and red and yellow. Okay, so red and yellow, two flower like two plants he has crossed. Then the result in this it has come is pink. Pink flowers has come. So that is why nowadays if you see in the garden, botanical garden or anywhere where different colors of plants and different types of plants which are with different colors, many colors has come. First there are very less colors, after scientist has done different. So the character which has shown the red and yellow, then it came pink flowers. Okay, so again codominance is, again in codominance, the definition I will tell you. In codominance, when the red and yellow crossed, then again same red and yellow only has come. So the definition of incomplete dominance is appearance of an intermediate phenotype, appearance of a intermediate phenotype. This is the one and this is the color 2. There is a between intermediate phenotype, physical character which is shown pink color, that has come in heterozygous condition. So that is the, even if you suppose not flowers, you take a curly hair father and straight hair mother. If you cross, the baby will get, if this is the this thing, wavy hair, daughter or son. This is the incomplete dominance. When two homozygous parents of different phenotypes are crossed, then it has come the intermediate phenotype. Like that, suppose for codominance, the definition is appearance of a mixed phenotype which is in heterozygous condition. So another dominance is complete, then red flower only. Complete dominance, for complete dominance, when red and yellow has crossed, then completely red color flowers came. That is called complete dominance. When curly and straight crossed, only curly generation has come. That is the complete dominance, which has a more this thing. They have shown that. So what all other parts of this lesson are? The next one, which is for multiple alleles, which are for blood groups. In this same lesson only, there are many topics are there, and most numbers are marks are 27 marks will be coming out of 70 marks. So there are practicals also on that. Even we are going to learn in this lesson pedigree analysis and different diseases of Mendelian disorders and chromosomal disorders, even chromosomal theory of inheritance, and the sex determination decided by different birds and insects. So the everything has decided by, suppose these are the chromosomes which are paired up. So these chromatids are called sister chromatids, same. Okay, in chromosomal theory of inheritance also, chromosomal theory of inheritance regarding chromosomes, Sutton and Boveri, chromosomal theory of inheritance. Sutton and Boveri, discovered by Sutton and Boveri in 1902. Okay, so here he has told they work out the chromosome movement during meiosis. So the chromosome meiosis and mitosis. The meiosis which is in reproductive organs, when suppose these when parents from male sperm and female ovum, so egg forms. This is a zygote. So when it is single celled, then multiple 41 meiotic divisions, then only these cells will 41 undergo. From first day of the ovulation, the single cell become different. As you know that from cell, different cells, tissues will form. Then from tissues, tissue system. Then from tissue system, organs, organism, organ system and organism. This is what, see, when it is single cell in a weeks of baby, slowly, one by one parts, how it is occurring. 41 meiotic divisions. It is a neat question which is repeating. Then the organs will slowly form. First cell, then many cells together, tissue in the body, which are like slowly eyes, hair, body organs which are internal organs and outside organs. Slowly by month, by 10 months, all organs are forming and complete baby has come because of this meiosis and mitosis, these divisions. So this is what chromosomal theory of inheritance, how the movement of chromosomes happen in these cells. The single cell inside nucleus will be there, the movement slowly, these divisions, metaphase, anaphase, those divisions happen, then the cell completely occurs. In Mendel has done research and chromosomal theory of inheritance. Afterwards Sutton and Boveri has done. This is what they have told, how movement of, they have observed several experiments. They have observed that the movement of chromosomes inside, how these are getting paired up and those organs are forming one by one. He has explained in this chromosomal theory of inheritance. So here also same, homologous chromosomes lead to segregation of a pair of factors they are carried. Even because of even one more question, Mendel's work on principles of inheritance was remain unorganized, one unrecognized. Why? And these are the questions are coming in the next class. I am going to explain about Drosophila, the further how the Drosophila and sex determination, these things I am going to explain. Thank you. And before that, please read one by one you are watching one by one video. Please read book, what available material you have. Understand, if you read once you might not get some concepts. Please read self-reading, and several times of reading will give you the best result. Keep on reading the tough topics, you keep focusing, and anyway whatever easy for you, you will score marks. When they don't leave any questions in the question paper without knowing. Think and write, attempt it. Obviously you will get the answer. So the next part I'm going to even tell